This talk will explain why the ocean's twilight zone demands our attention. My name is Adrian Martin, and although I am based at the National Oceanography Centre in the UK, I'm giving this talk on behalf of the Joint Exploration of the Twilight Zone Ocean Network, recently declared as one of the programmes under the United Nations Ocean Decade. To start with, I'm going to explain why we should care about the twilight zone in the first place. We already know that the twilight zone contains a huge number of organisms. Just in terms of the mesopelagic fish, recent estimates put their total biomass at around 1 to 20 billion tonnes. That makes some of these fish, in the same family as this elongated bristlemouth shown here, amongst the most abundant vertebrates on Earth. For comparison, surface waters of the ocean hold around 1 billion tonnes, and the combined weight of humans on Earth is around 500 million tonnes. The twilight zone also contains a huge diversity of organisms, all the way from bacteria, some of which colonise the sinking material, through zooplankton, fish, all the way up to iconic species such as sperm whales and their prey such as this giant squid. We also know that the twilight zone is important in terms of taking carbon out of the atmosphere and storing it deep within the ocean. This is through a combined mechanism known as the biological carbon pump. However, it is rather more complicated than that simple name suggests. This recent paper by Boyd and co-workers breaks it down into its component pumps. All I really want to highlight here is the fact that the full span of organisms occupying the twilight zone plays a role in that transport of carbon to depth, all the way from dead phytoplankton at the surface through bacteria to fish and larger organisms transporting carbon directly to depth. The twilight zone is also a critical region for the recycling of nutrients, which sustain primary production in the surface. These plots show results from a model run for me by Francisco de Melo Verissimo. What they show is the consequence of changing the rate at which organic material is recycled within the twilight zone. The value B corresponds to how rapidly organic material is recycled. A small value of B means that it is recycled slowly, and so a lot of material escapes, and so you can see small values of B result in a large amount of nutrients at depth. Corresponding to that, you can see that small values of B also give smaller primary production. What is happening is that the twilight zone is less efficient at recycling the nutrients, more nutrient ends up at depth, and global primary production drops as a result. The twilight zone is also experiencing the impacts associated with climate change, as is the rest of the Earth system. We're very familiar with many of the impacts being suffered by the twilight zone: changes in temperature, acidification, and, as I've shown here, oxygen. This plot, from a recent review by Andreas Oschliers and co-workers, shows the changes in dissolved oxygen between the surface and 1,200 meters over the 50-year period from 1960 to 2010. You can see that there is a widespread and significant decrease in oxygen, and this is important because it has a direct effect on many physiological processes taking place within the twilight zone. More recently, deep-sea mining has also emerged as a potential impact on the twilight zone. It may be thought that extracting minerals from the sea floor at 5,000 or 6,000 meters depth would have little impact, but one of the potential consequences of this is the release of waste into the mid-water, or if there's mining taking place at shallower depths on seamounts, for example. I've already mentioned that the twilight zone is home to a huge number of fish. That abundance carries its own threat and may lead to commercial fishery of those organisms. At the moment, the economic viability of such a fishery is uncertain. However, something to be aware of is that in addition to the impact of the fishery itself, which is still largely unknown, there is an additional threat from the combined impact of fishery plus climate change. These are some very preliminary results from modelling work done by Douglas Spears and Emma Dolmer under the EU MESO project. What they're showing is the modelled abundance of a Benthosema species fish. That's one of the mesopelagic fish that I mentioned earlier. You can see, even though there's a lot of variability from year to year that I'm not showing here, there is a significant decline in the abundance between 1988 and 2050. In the model, that largely results purely from changes in temperature. What we don't know is how any fishing pressure on top of that would further exacerbate that decline. An area of increasing interest is that of carbon dioxide removal. This is the process of removing carbon dioxide from the atmosphere and hopefully averting some of the worst consequences of climate change. A number of fora are springing up to discuss processes that might allow us to do this. This is just one, oceancdr.net. There are a number of others I'm sure you can find on Google. I put this one up just to highlight the fact that 4 of the processes that they talk about as worth discussing would have direct impacts on the Twilight Zone, either by changing the conditions or formation of material in the surface waters that feed the Twilight Zone, or by directly affecting the transport of material and water through the Twilight Zone. There is an ongoing United States National Academies of Science, Engineering and Medicine review into these processes. The Twilight Zone is, in the strongest use of the term, international. The top plot shows in dark blue areas beyond national jurisdiction. The lower plot shows bathymetry. The continental shelf is shown as orange, and you can see that the areas beyond the shelf, where the Twilight Zone occurs, strongly correlates with the areas beyond national jurisdiction. Hence, our approach to the Twilight Zone and our behaviour in the Twilight Zone needs to be discussed in an international forum. I'm now highlighting 3 questions that I think we need to tackle for the Twilight Zone, particularly over the coming decade. How many organisms are there, and how diverse are they? How is the organic material within the Twilight Zone being modified? And what are the pathways into and through the Twilight Zone? For all of these questions, there are many things that I could discuss. Because I have to focus on just a few, please do regard them just as what they are, examples, and I would strongly encourage you to look into the whole array of things going on in the Twilight Zone. As the first example under this question of how many and how diverse, I want to touch on optics and machine learning, which I think has been a revolution over the course of the last few years. On the left-hand side, I'm showing images taken with the CPIC sensor developed by Coastal Ocean Vision. In addition to shape and size, it also gives colour, providing an extra dimension in our recognition, classification and study of the organisms in the Twilight Zone. On the right-hand side, I'm showing images taken with the UVP. This is from a paper by Trudnosket et al. In Nature Communications just this year. They have gone beyond trying to recognise organisms and are now confronting the difficult task of recognising the very variable marine snow, the dead material sinking down through the Twilight Zone. This is known to display a huge variety of shape and texture, and so what they have done is to apply machine learning to try and classify this marine snow, and I'm showing results here of breaking it into 5 groups. And I think this marrying together of optics and machine learning is hugely promising for future study, particularly of the smaller organisms and entities within the Twilight Zone. And I would refer you, for example, to the ITAPINA initiative ongoing, bringing together people looking at optical techniques. As my next example, I want to go further up the food chain to fish and discuss acoustics. I'm going to be using some preliminary data given to me by Roland Proud and Andy Brearley, who are analysing data collected as part of a Spanish cruise in 2019 as part of the EU Summer Project. We know that there are a huge number of fish within the Twilight Zone, between 1 and 20 billion tonnes. However, 1 to 20 clearly represents a large uncertainty, and one of our main priorities for the next few years is going to be reducing that uncertainty so that we have a more precise idea of the number of fish within the Twilight Zone. One of the main tools for doing that will be acoustic methods, but calculating fish biomass from acoustics is no trivial thing. On the left-hand side is a schematic showing the varied community of organisms within the Twilight Zone that undergoes a daily migration between depth and the surface across the Twilight Zone. This is one of the largest animal migrations on the planet, taking place every day. In the middle is the signal recorded by a ship's echo sounder, and you can see a clear signal associated with this migration. However, turning that acoustic signal into an estimate of biomass is rather difficult, not least because there are organisms within the Twilight Zone that exhibit similar acoustic responses despite being rather different organisms. For example, There are siphonophores which exhibit a similar acoustic response to fish with gas bladders. We can augment that data collected from ship echo sounders with more focused echo sounder data from lower devices, such as the one that Andy and Roland have been using. But in addition, we need to corroborate these acoustic methods with more direct identification of the organisms, initially being done through pelagic trawls, but in the future using techniques such as environmental DNA and camera approaches. This multiple approach will be key to reducing the uncertainty on our estimates. The next question I want to touch on is, how is the organic material in the twilight zone being modified? As my first example on this question, I want to look at organism interactions. On the left-hand side, I have some video footage of experiments carried out by Thomas Kirby et al. We're looking at Acartia copepods feeding on prey. It's now thought that these organisms play an important role in the attenuation of the flux of aggregates sinking through the twilight zone by breaking it up into smaller parts. This doesn't just slow down the transfer of organic material through the twilight zone, it also potentially impacts the ecosystem itself. For example, there is a microbial gardening hypothesis that breaking up the aggregates fosters bacterial growth on the smaller aggregates. Going up in size, this is the mesobot, and you can see this in a recent paper by Dana Yerger and co-workers in Science Robotics. This is an AUV, an autonomous underwater vehicle, which has been designed specifically to follow mesopelagic organisms in their own environment, allowing them to video interactions between organisms in situ. The next example is once again looking at the gravitational flux, the sinking flux of aggregates, marine snow, out of the surface into the depth. And I do this to illustrate the potential importance of the biogeochemical Argo array rapidly being expanded to biogeochemical questions particularly relevant to the twilight zone. This is some work done by Nathan Briggs and co-workers, appeared in Science just last year. Using fluorescent sensors and backscatter sensors on these floats, you're able to tease apart, first of all, the signal of sinking material. You can see that in these traces here of fluorescence and backscatter extending in these diagonal lines on these time versus depth plots. But also you can break that signal apart into large and small components on the top and on the bottom. And then you can go one step further and you can look at the rate at which those two are being attenuated and use it to infer the rate at which the large particles are being broken into the smaller ones. So using this distributed network of floats, we can now start to look at the mechanisms responsible for breaking up this flux of sinking material. The third question is looking at pathways into and through the twilight zone. For the first example, I want to reinforce the fact that we do not live in a one-dimensional ocean. And in order to understand what's happening with the twilight zone, we need to understand it in a fully three-dimensional framework, acknowledging the full role of the ocean circulation. We know that there are a number of physical pathways into the twilight zone at scales ranging from the submesoscale of a few kilometers to the basin scale. What I'm focusing on here is the potential role of the smaller scales, the submesoscale. We've known for some time that you can have very strong vertical circulations at the submesoscale. On the right hand side, this is results from a modelling paper by Laura Resplondi et al. But we have similar observations showing this effect. What you have are strong vertical velocities, you can see in the lower plot, associated with this plume of particulate organic carbon being taken down to 500 meters, well within the twilight zone. So we've known that these sort of processes take place for a while. The open question is, how significant are they? That example from the Resplondi paper goes on to be used in a fuller context where they argue that the cumulative effect of these submesoscale subduction events is actually quite small in a global sense. Go back a few years and Melissa Romand and co-workers in Science are using a more observational approach where they estimated, as you can see in this figure, that the flux due to these subduction events could actually be a very significant part of the passive flux, that's the gravitational flux. So to my mind at least, it's still an open question on how important these small-scale physical processes are. As a second example on this question, I want to look at what's been termed the active flux, and in particular the role played by fish in moving carbon through the twilight zone. There's been an ocean carbon biogeochemistry working group looking at this for the last few years, and in particular they've looked at the contribution of fish to this flux and how that contribution varies with depth. One potential being that the relative contribution from the flux of fish moving carbon to depth actually increases with depth, effectively because once the carbon is inside the fish, it cannot be attenuated on the way down, and so if the fish travel to depth and release it as fecal matter, then potentially that is a much more efficient way of transporting carbon to depth. There aren't many studies, and they admit that in the paper, but putting together the results that are out there, they would come up with an estimate that the mean fish contribution to the total carbon flux, this is leaving the surface and going into the twilight zone, is around 16%. And that's equivalent to an annual carbon flux out of the aphotic zone of about 1.5 petagrams. But please do bear in mind the fact, as I've described, that contribution to the total flux may increase with depth. So, hopefully those three questions have given you a taste of what we need to tackle regarding the twilight zone and understanding its function. I want to finish by stressing the need for collaboration in doing so. As I've already said, understanding the twilight zone is going to be a huge endeavour and is going to require people collaborating from across countries and disciplines. I think a nice example for this is the workshop that there was the very start of Jetson. There was a workshop in 2019 that brought together everybody from individual PhD students to people representing multinational projects. We already have collaborations emerging as a result, and I see this as being the pattern for tackling the twilight zone in the future years. Collaboration will be critical within and across disciplines: theory, lab work, field work, modelling. However, it's always nice to have a big finish, and so as a grandstand finale, I want to use this spectacular example of collaboration that took place this summer. To finish, I'm not going to give you a list of conclusions. Instead, I'm simply going to encourage you to get involved. This marks the start of the United Nations Ocean Decade. Those who are PhD students now may be professors by the time that the decade closes. So far, Jetson already runs monthly open calls. All people are welcome, all contributions, and the slides and recordings from previous ones are held on the webpage. We run a GitHub page to share data, methods, and cruise opportunities. So please look there if you're looking for a chance to go on a cruise, and please, to those who are running cruises, put spare berths there so that people can be aware of them. Within Jetson, we already have 24 projects and counting. Over the course of the decade, we have lots more activities planned, and clearly more is possible. You can influence what that is, so please do get involved. Thank you.